Hello, everyone, and welcome to today's webinar. From Transcriptomes to Epigenomes, Introduction to Single-Cell RNA-seq and Single-Cell ATAC-seq. I am Israel Gonzalez of LabRoots, and I'll be your moderator for today's webinar. Today's educational web seminar is presented by LabRoots and is brought to you by Bio-Rad. For more information on Bio-Rad, please visit biorad.com. That's bio-rad.com. Before we begin, I would like to remind everyone that your questions are welcome. You can submit them into the Ask a Question box on your screen, and our speakers will be available to answer as many as they have time for following the presentation. You may also use the question submission box for any technical issues, if any may arise. I'd like to now welcome our speakers, Errile Pusod and Patricia Schnepp. You can open to view our speakers' complete biographies on your screen to find out more about them. Errile and Patricia, take it away.
Thank you very much for the very nice introduction, and thank you everyone for joining today's presentation. Again, my name is Errile, and today's webinar is the first in our series of educational webinars on single-cell sequencing. In this webinar, we will be discussing the basics of single-cell sequencing, specifically giving you an overview of transcriptomic and epigenomic profiling. Next, we will introduce the Bio-Rad single-cell next-generation sequencing solutions, our library preparation kits, and show you some interesting data on case studies and applications. Finally, we'll leave some time at the end to answer some of your questions. To get started, let's go back to the beginning, the central dogma of molecular biology. This theory demonstrates how genetic information flows from DNA to RNA in the transcription process and how RNA is translated into its final product as a protein. Each of the molecular biology research can be categorized in what is pertained in the suffix omics, where the big four omics are genomics, epigenomics, transcriptomics, and proteomics. The genome encompasses the complete set of an organism's DNA, while the transcriptome only looks at what portion of that DNA is transcribed or what genes are expressed. Epigenome, on the other hand, focuses on the factors that regulate the expression of those genes, either they are repressed or induced. And finally, proteome looks at the final protein product and its functions. In today's webinar, we will be focusing on the transcriptomes and epigenomes. Traditional methods of these omic studies were done in bulk, where the information that you're getting is an average expression values of all the cells in the sample. Now, this is not a problem if there is only one cell type in the entire sample, but because biology is so complex, that is rarely the case. Enter single-cell sequencing, where information is generated from individual cells within the sample. This allows researchers to not only characterize various cell types in a heterogeneous population, but also helps researchers identify rare cell populations or even discover new cell types. A really popular and nice analogy for that difference is the difference between a fruit smoothie and a fruit salad. The image on the left, the fruit basket that you see, represents a complex tissue, and each of those fruits is a cell type. In a fruit smoothie, representing bulk sequencing, it's very difficult for us to know what fruits were blended up and how much of each fruit was used. That smoothie may look very red, but may contain only a single piece of strawberry that's very pigmented. There might also be an apple in there, but the taste is masked by the strong flavor of banana. However, in a fruit salad, representing single-cell sequencing, you can clearly identify what fruits were used to make it and how much of it was used. We can even take it a step further and characterize each fruit based on its flavor, its texture, its nutritional content, and even eliminate the fruits that we don't want. The first ever single-cell sequencing study was published in Nature Methods in 2009 by Tang and others on a 4-stage murine blastomere. Since then, there has been a huge surge in the number of published papers that come along with the advancement of the technology. This is a number that continues to grow to this day. This was made possible by the key technological milestone of next generation sequencing, and something that would take us forever if we continue with the traditional Sanger sequencing. The graph that you are seeing shows us how much it costs to sequence 1 million bases of DNA, which back in 2001 would cost us $10,000. And fast forward 21 years later, the same number of bases would cost us a cent. With the advancement also of all of these single-cell sequencing technologies, various methods were also developed. The first ever single-cell sequencing paper in 2009 that I mentioned previously is the one on the very left, where it was done manually and in exactly one cell. Now, I'm not going to go through each of these methods, but some of the most commonly used ones are the plate-based methods, where it utilizes flow cytometry to sort individual cells into each well of a multi-well plate. This method includes a lot of manual work, and so it tends to be pretty costly. And in terms of throughput, it can only accommodate up to a few hundred cells. In situ barcoding, on the other hand, does not isolate cells individually, but instead cells are fixed and either the mRNA or DNA are manipulated with multiple rounds of barcoding. And those multiple rounds of barcoding are what adds the unique tags to identify each cell. This method is fairly inexpensive because there is no need for any special instrument, but it can be labor intensive and it can span multiple days with a variety and along multiple pipetting steps. The droplet-based method, however, is a method that uses microfluidics that creates millions that are commonly called droplets. And in these droplets is where a single cell is encapsulated and where the chemistry occurs. This method is more cost-effective and it has higher throughput because you can encapsulate tens of thousands of cells in a single experiment. And it's much suited for larger scale studies because you can simultaneously run multiple samples at the same time. Because of the wealth of information that can be generated using single-cell sequencing, its application also extends to a vast area of research that improves our understanding of complex biological systems. Bio-Rad single-cell next-generation sequencing solutions are droplet-based methods composed of the single-cell 3' RNA-seq, the single-cell ATAC-seq, both of which are compatible with the DD-seq single-cell isolator for droplet generation, and the accompanying open-source omniscient analysis software that Patricia is going to touch more on later. The DD-seq single-cell isolator is a small footprint automated instrument that partitions single cell on a disposable cartridge in only 5 minutes. It can accommodate up to 4 samples that are processed simultaneously with 60 to 90% capture efficiency and applicable for use with larger cell types. Now let's go through the 2 cost-effective library preparation kits that we offer. We have the DD-seq single-cell 3' RNA-seq kit that is used for transcriptomic profiling, and the DD-seq single-cell ATAC-seq kit that is used for epigenomic profiling. Both kits allow for a single-day workflow, if you so wish, but it can also be broken down into several days or several weeks depending on your experiment schedule, as it has multiple stopping points and storage conditions can vary depending on your preference. You can keep your samples in the fridge for short-term storage, usually about 24 hours, or in -20 for longer-term storage. The performance of both of these kits is on par with what's on the market today, and it can support cell types of larger size with expected outputs of over 5,000 cells per sample on the higher end of cell loading. Specifically for the single-cell ATAC-seq kit, we also support nuclei or permeabilized whole cell as input, depending if you're using samples that are more resistant to lysis or if you're using fragile cells that require gentler handling. That is possible because we have validated 2 different tagmentation protocols for those. We have the OmniATAC tagmentation protocol, and this tagmentation protocol is validated for different cell lines or any other cells, again, that are resistant to lysis. And so this one uses a much harsher detergent so you can have efficient cell lysis. But we also have the whole cell tagmentation protocol that was validated for PBMCs, for bone marrow derived mononucleated cells, or other primary cells that requires gentler handling. Now, to go through each of these kits and how they work, first, let's look at the DD-seq single-cell 3' RNA-seq. Now, in the next few slides, I'm going to play a video to see how the technology works. The single-cell 3' RNA-seq kit profiles gene expression from each cell by encapsulating individual cells in droplets within 5 minutes. Beads are overloaded in each droplet, but ensuring that a single cell is encapsulated. Within the droplet, mRNA is captured in the 3' end by the poly-T oligos contained within the beads. We also have a unique deconvolution oligo, the one that you are seeing in blue, and this allows us to link multiple beads that are coming from the same droplet. This cool process is done informatically, and this will be explained further by Patricia in more detail. Next, we have the DD-seq single-cell ATAC-seq kit that profiles cells based on their epigenome. And then again, in the next few slides, we'll show a video of how the technology works. So the word ATAC in single-cell ATAC-seq stands for Assay for Transposase Accessible Chromatin with Sequencing. The single-cell ATAC-seq profiles epigenome based on the open regions of the chromatin within the chromosome, which is often associated to sites where active transcription is happening. Off with cells that are either permeabilized or where nuclei is directly isolated. In these open chromatin regions, we have a TN5 transposase enzyme that allows for the tagmentation or the cutting up of the DNA in random sequences that allows for the addition of adapters. Transposed nuclei then are encapsulated individually using the DD-seq single-cell isolator within 5 minutes. The addition of these adapters make it compatible for generating sequencing-ready libraries. Bioinformatically, these sequencing reads are mapped to reference genome to reveal a genome-wide accessible regions. Next, I will turn this over to Patricia to talk about our unique deconvolution oligo technology and give an overview of data analysis and present some cool data.
Thank you for that, Errile. The deconvolution methods used by both DD-seq single-cell assays is unique to our system. In this slide, you can see our DD-seq single-cell isolator and its internal microfluidics mechanism. In the first 2 channels flows either individual cells or tagmentated nuclei, and the other channel flows barcoded beads and other reagents. These mix and flow through oil to be encapsulated into individual droplets. However, the flow rate and concentration of the first 2 channels determines the probability of how many cells and beads are in each droplet. When cells or beads are kept in low concentration, they are encapsulated in mainly a single object manner. A cell load of 1,000 to 10,000 cells keeps the Poisson distribution lambda low, ensuring only one cell is in any one droplet that is formed. A higher lambda value could result in droplets containing more than one cell, which would not allow us to analyze these cells individually. When you keep the concentration low for both the beads and cells, you were able to ensure in any one droplet, multiple beads or cells are not present. However, this double Poisson distribution also results in there being an even lower probability of both one cell and one bead being encapsulated in the same droplet. Thus, cell utilization is below 10%, and much of the sample is lost. To overcome this issue, we use a method to overload the beads while maintaining a low lambda value for the cells. This ensures that there is still only one single cell per droplet. However, this creates a bioinformatic issue in determining which beads, since multiple beads can be encapsulated in the cell, in the droplets now, and how many beads are present in that single droplet. To overcome that issue, we use the unique barcodes that each bead is given to recreate bead networks. Each bead network is made up of the beads that were encapsulated in each droplet. Once a bead network is determined, all the genomic or transcriptomic information contained in all those beads are grouped together to recreate the full cell that was encapsulated in that particular droplet. To recreate these bead networks, you use a slightly different strategy for each single-cell assay. For the DD-seq ATAC-seq kit, we identify the start and stop positions of each sequencing reads for each of the beads. These positions are then compared to the start and stop positions of the sequencing reads of all the other beads in the sample. The beads with a high overlap of these sites are more likely to be from the same droplets compared with beads with low or no overlap. Once these full comparisons are completed, the resulting bead networks can be determined, and the genomic information for these connected beads can be brought together to identify the full genomic information from the original encapsulated cell. In the DD-seq 3' RNA-seq kit, we use those unique capture sites called deconvolution oligos, or DOs. These DOs only bind to the DOs from other beads. Each DO sequence is linked with that unique bead barcode also found on the mRNA capture oligos. After binding to DOs from other beads, the full read will contain the unique barcode from the first bead with the unique barcode from the second bead. After sequencing, our companion bioinformatics software is able to identify these unique DO reads. By identifying which bead barcodes were hybridized by these DO sequences with barcodes from other beads, the software recreates the bead network of which beads were encapsulated in the same droplets. Thus, all the transcriptomic information of these connected beads can be brought together to identify the full transcriptomic profile of the original bead. By using this method of overloading beads, we obtain a cellular utilization of 60% or more across many different number of input cells, which is higher than that 10% cellular utilization that normal double Poisson distributions would obtain. In some cases, we can reach a cellular utilization of 70% or more. Following cellular partitioning and library preparation, these libraries are sent for sequencing, returning FASTQ files that are used as input for our companion software, the Omnition analysis software. Omnition can analyze data from both the DD-seq ATAC-seq kit and the DD-seq 3' RNA-seq kit. Omnition is part of the secondary analysis in terms of the hierarchy of NGS data analysis. Primary analysis is the process sequencers perform to identify the exact base order of individual reads in a library. The outputs from primary analysis are the FASTQ files. Omnition performs secondary analysis, including quality control steps of the raw reads, alignment to the reference genome, debarcoding and deconvolution of those bead networks, and creating assay-specific metrics to help in determining the quality of your sample, and creating tertiary-ready output files and a summary report file. Tertiary analysis includes analyses such as cell type annotation, differential expression, or differential accessibility analysis. Omnition's output files can be used as input files for many open source and commercially available tertiary analysis tools. Omnition is that companion software for our current 2 assays, but also for future single-cell assays Bio-Rad will be releasing. It can run on a Linux system using single command line execution. If you have multiple samples using the same reference genome, you can do batch processing of these samples together in one Omnition run, which reduces the hands-on time necessary for secondary analysis. Omnition provides tertiary-ready analysis output files unique to each sample, even if run in a batch run. A version of Omnition will be available in Bio-Rad's cloud computing website in the near future. Once you have finished your secondary analysis, Omnition provides a report containing a summary table and initial quality control metrics so you can assess the quality of your sample. One such QC graph is the barcode rank plot. This plot takes the deconvoluted droplets and orders them from the largest number of fragments or genes to the smallest. By doing this, you will find a flat start in the graph with a curve or knee, followed by a cliff. The cliff indicates separation between droplets containing genomic or transcriptomic information from true cells, those droplets containing cell-free molecules or other background noise. Omnition determines the reflection point to determine the cutoff between the droplets containing two cells and those containing background. Lowering the inflection point will increase the sensitivity of your cell calls, but also increase the risk of including background DNA or RNA. Raising that inflection point increases the accuracy of calling true single cells and reduces the risk of including background DNA or RNA, but you may be losing true cells that contain a lower genomic or transcriptic load in your sample. Omnition additionally provides information about the number of unique fragments or genes per cell in your sample, as well as the number of reads per cell. These metrics can be used to help evaluate your sample. Lastly, mitochondrial proportion can be used. If the majority of a cell's genomic or transcriptomic load is from the mitochondria instead of the nuclei or cytoplasm, this suggests that cell burst prior to encapsulation, and only the mitochondria was captured in the droplet. These droplets may be removed during tertiary analysis from your dataset. There are a few more ATAC-seq specific metrics you can use, including the transcriptomic start site enrichment score. You would expect the majority of your reads in your sample to primarily be around the TSS sites in the genome, which is what this metric measures. You can also look at the insert size distribution for the expected nucleosome periodicity of decreasing peaks about 200 base pairs apart. Lastly, you can look at your FRIP score for the fraction of unique reads mapped to the identified chromatin region. The final metric Omnition provides is the multiplet rate. This is calculated when you are running a sample that contains two reference genomes. In this example, mouse cells were mixed with human cells. We can measure how many reads align to the human or mouse genome in each droplet. The vast majority of our reads will align to either one of the genomes. For those few droplets, you will find reads aligning to both genomes. This is indicative of a droplet containing at least one human cell and at least one mouse cell. These multiplets should be removed during tertiary analysis. These multiplets are due to the method of encapsulation, but occurs at a very low rate. Finally, I would like to discuss a few use cases of the DD-seq, ATAC-seq, and DD-seq 3' RNA-seq assays using real samples. The first is two PBMC samples run on either the DD-seq single-cell ATAC-seq kit or the DD-seq 3' RNA-seq kit. Both were sequenced according to their respective protocols and analyzed using the Omnition software. After quality control was performed, we were able to perform cell type annotation, including cell types that were found in both samples, such as B cells, T cells, natural killer cells, and monocytes. With this multi-omic data, we can look for differential accessible peaks and differential gene expression that suggest a genomic relationship between a gene in a particular cell type, or of genes where the differential gene expression is not due to the changes in the gene's accessible chromatin. For example, in the CD14 monocytes, we find a chromatin peak specific for the gene EREG. This correlates with an increased expression of EREG in the 3' RNA-seq data. This suggests a genetic relationship or genetic regulation of the EREG in CD14 positive monocytes. With this type of multi-omic data, you can do more of these type of analysis and other analysis. Furthermore, we have run multiple other sample types beyond normal PBMC cells using our DD-seq 3' RNA-seq kit. This includes prostate cancer, iPS cells, derived neuronal cells, primary skin cells, and HIV-infected PBMC cells. All of these samples have a cell utilization between 60% to 78%. Additionally, we captured a number of genes and UMIs of expected values for each sample type. Since each sample type has a different transcriptomic load, we expect the number of genes and UMIs to be sample-specific. Let's take a closer look at one of these samples, the iPS-derived neuronal cells. In this experiment, microglia was induced to become iPS cells, then transformed into neuronal cells. In this experiment, a gene of interest was knocked down and then transformed into the neuronal cells. Using our DD-seq 3' RNA-seq assay, we can investigate the role this gene of interest plays in the transformation process into neuronal cells, including how it affects the types of neurons in the final sample. When we visualize the sample in our UMAP, we can see areas of more blue, or areas with more knockdown cells, and areas with more orange, or more control cells. To investigate this further, we computationally calculated clusters in this combined sample dataset and counted how many control and knockdown cells were in each cluster. Using these cluster cell counts, we can calculate the percentage difference between the two sample types in each cluster. A more negative percentage change indicates more knockdown cells in the cluster, and a more positive percentage change indicates more control cells in that cluster. We can find cluster 2 and 8 containing a high percentage of knockdown cells, in clusters 1 and 4 containing more of those control cells. When we look at the UMAP, clusters 2 and 8 are next to each other, and clusters 1 and 4 are next to each other, suggesting these clusters represent potentially similar neuronal subtypes that are either enriched or downregulated by the expression of our gene of interest. Further analysis could be done to determine which neuronal subtypes are in these clusters, as well as gene expression trajectories that are regulated by our gene of interest. Finally, I would like to highlight a publication of a benchmarking study of multiple single-cell ATAC-seq kits, including the DD-seq single-cell ATAC-seq kit. This paper compared the performance of our DD-seq ATAC-seq kit to many other assays and found our kit had a high TSS score and a high FRIP score compared to other assays, which is a good indicator for a good signal-to-noise ratio using our assays. Lastly, we have many additional resources on the Bio-Rad website. We have the user guides for both of our assays, as well as Omniscient, a sample preparation guide, and a published publication list that is updated with new papers using one of our single-cell assays. To find these resources, you can use the bulletin numbers listed here to search the Bio-Rad website, or go to www.biorad.com/ddseq for direct links to these documents and many more. And with that, on behalf of Errile and myself, I would like to thank LabRoots and everyone here for having us for this webinar. Thank you.
Well, thank you both for that informative presentation. Let's go ahead and dive into the Q&A segment now of the webinar. And to our audience, I'm going to continue to encourage you to submit your questions for our speakers. We already have several coming in. So let's go ahead and start with this first one. I believe this is going to be for you, Patricia. It says, hello, I've been trying to process data sets that contain one control and one treated scRNA-seq and SC ATAC-seq data. What I want to do is compare peaks between one cluster in the control and the same cluster in the treated. I was wondering if you had any comments on which computational tool to use. So Patricia, I'll let you take it from there. There's a little bit more to the question, but I'll let you kind of take it now.
Yeah, this kind of work I typically do with SIGNAC. Now with SIGNAC, you would need to bring the two samples together, and then subset that Seurat object on the cluster. And then you can use that find markers function, but you to change the indents into looking at the treatment option and not cluster, because it automatically goes to cluster. And then you should be able to do this type of comparison in the single cell ATAC-seq data. It's maybe not super clear, but once you go deep into those functionalities and the different options that you can do in the different functions like find markers, you can do this type of analysis.
Perfect. Thank you so much for that. Our next question is, and I'll let either of you kind of jump in here. What is the starting number of cells required for each technology?
Yeah, I can answer that question. And so for each technology, they are kind of different in their workflows. If we start with the single cell RNA sequencing, the input that we recommend, it has a range, and so at the lowest range it's 1,000 cells, and at the highest range it's at 10,000 cells. Now keep in mind that this is at the stage where we are loading the cells, and so you want to take into account all of the treatments that you would want to do with your cell to make sure that they are of the highest quality, that they are viable. If there are any other treatments like removing red blood cells, that cell removal, or any other enzymatic treatments that you want to do, you want to make sure that you have, before cell loading, enough cells that you need for that. And so recommended, we would say about like 150,000 cells to 300,000 cells. With the single cell ATAC-seq, that's a little bit different because, like I mentioned, this one, we have a separate step for nuclei isolation and tagmentation. And so with that, with the cell or the nuclei loading being at like 500 to 10,000, you want to make sure that you have enough cells for the tagmentation process to happen. And with that, we require 60,000 cells, and so, or nuclei. And so if you needed 60,000 cells or nuclei at the tagmentation step, you want to make sure that you have enough in the upstream process to make sure not only that you have enough cells, but also you're seeing a pellet, right? Because it also depends if you are a first-time user or an experienced user. That can vary in any process that you do, in any treatment that you do. You are expecting any cell loss, so making sure that to keep that in mind.
Perfect. Thank you. Our next question is, how does the deconvolution process differ between single-cell RNA-seq and single-cell ATAC-seq?
Yeah, so I'll go ahead and talk about that one. So in this one, the deconvolution process is slightly different for the single-cell ATAC kit and the 3' RNA-seq kit. In the case of the single-cell ATAC-seq kit, we're looking for those sequencing reads that contain the same start and stop site positions between two different beads. And then for one bead, we'll get all those start and stop positions and compare that to all the other beads that are in the sample. And we'll count how many reads are matching and how many that aren't. And to kind of give a number to that, we're using what's known as a Jacquard index similarity score. So the higher the Jacquard score, the more reads are shared compared to that are unique. If it's a low score, more of the reads are not unique compared to being shared. So those beads, two beads that have a very high Jacquard index, means that a lot of their reads share the same start and stop positions. That they're more likely come from the same droplet and can be brought back together to recapitulate the original cell. And we can do that if there's 2 beads in a droplet, 3, 4, as many beads are in the droplet. We can use this Jacquard index to help determine which ones were in the same droplet and which ones weren't. In the case of the 3' RNA-seq, we use the deconvolution oligos that are unique to each of those beads. So those oligos are the capture sites that are unique to the other capture sites on other beads, but aren't going to align into the genome. So we're only going to be connecting the barcode from one bead to the barcode of a second bead. And we can sequence a lower number of those reads to be able to rebuild the entire bead network compared to the number of reads you need to look at, get the gene counts from the transcriptome. And once we have those deconvolution reads, then we can build back the bead network. And both of these different deconvolution strategies are done by Omnition, the companion software, so a customer or user doesn't have to do that themselves.
All right, thank you for that, Patricia. Let's go ahead and go to our next question here. How does the tagmentation enzyme work?
Sure, I can answer that one. So tagmentation is a reaction that occurs in both single-cell RNA-seq and single-cell ATAC-seq. In single-cell RNA sequencing, it is a process where the cDNA is fragmented into smaller pieces as part of a regular library preparation, so you can add adapters to make it sequencing ready. But I'll focus my answer on the single-cell ATAC-seq because the tagmentation process in this technology is where a TN5 enzyme cuts up chromatin DNA in fragments to allow for the addition of the TN5 adapters. And this process is random, and it gives a unique TN5 that gives information of nucleosome positioning, which is a key factor in gene regulation.
Errile, thank you so much for that. All right, moving right along, and as a reminder to our audience, you're more than welcome to continue asking your questions in the Ask a Question box beneath the slide window there. Is there a limitation in the size of the cells that we can encapsulate in the droplets?
I can answer that question too. So yes, there is. So, like because we are working with a microfluidic technology, we are limited in the size of the droplets and in the process of how it flows through the microfluidic channel. This is based on the cell's diameter and also based on a cell's deformability, how it's able to like squeeze into that droplet. Internally, we have data to show that we are able to encapsulate adipocytes as large as 50 microns. But as we are working with this product, with more and more customers are using it, we are expanding the sample types that we're using. And so coming up, we will be like having sample types of larger cells. We'll have data on those in the future.
Awesome. Thank you so much. All right. Moving right along. Can you get information about immature and mature RNAs from Omnition?
Yeah, so I'll go ahead and take that question. So indirectly, you can look at immature mRNAs from Omnition. So there is a parameter in Omnition that allows you to either include or exclude the intronic regions of the genome. So in that case, if one of your reads were to align to an intron, it may or may not be included in the final gene counts based on this parameter. The default for Omnition is right now to exclude those intronic reads from the final gene count. Thus, the information you're getting is more from the exonic reads that are found in those mature mRNAs out in the cytoplasm. However, if you wanted to include gene counts from the introns, you can go into the input YAML file, which is a YAML file that is used to put all of the information, including parameter settings, when you go to run Omnition. And so you can say to include introns in your final gene count in that YAML file. And so the final gene counts that you'll get will include information both from the mature and immature RNAs. So if you were to look at the RNA counts from the reads that excluded introns compared to the gene counts after you excluded the introns, that difference can give you some insight to potential immature mRNAs compared to more the mature mRNA counts in your sample.
Awesome. Thank you for that, Patricia. Going back to droplet size, we have another question here. What are the droplet size, or what is the droplet size for the two assays?
I can take that question. So the droplet size for the two assays is very similar. It is about 90 microns in size.
Perfect. Short and sweet. I love it. We have just a few more questions here, so let's go ahead and move on. Are the single-cell RNA-seq and single-cell ATAC-seq available as an integrated multiomic workflow?
Yeah, I'll go ahead and answer that one. So currently, the single-cell ATAC and single-cell 3' RNA are two separate kits, though we are working on an integrated workflow for the future, so stay tuned for information on that. But currently, because they are two separate workflows, you aren't able to take the same sample and look at the transcriptome and genome of the exact same cell. However, bioinformatically, we can bring those two assays together to still harness important information from a multiome view by still having those single omic data sets to begin with. And there are different computational tools out there that allow you to do that, as well as different functions in the well-established tools that a lot of people use to be able to do that type of analysis.
Great. Thank you very much. All right, we're getting an inflow of some questions here, so let's move right along. Let's see here. Let's go to, are you isolating RNAs from frozen cells? Someone wants to know.
Yeah, I can answer that one. No, so RNA is not isolated in these technology because what you are encapsulating is cells. So for the single cell RNA sequencing, the cells are encapsulated into the droplets, and the RNAs or the cell is lysed within that droplet. So you can make reverse transcription from the 3' end or from the mRNA of the cell.
Great, thank you so much for that, Errile. It looks like someone wants to some clarification here about the droplet size being smaller than 50 cell size. I believe that. I'm not sure if you can see that question, Errile.
I do.
Maybe you can clarify.
Sorry if what I said was not I didn't enunciate, but it's 19.
Perfect, thank you. All right, we have time for some a few more here. If I want to find an MOA of a drug, which kit would you recommend to evaluate the mRNA expression profile?
So I can talk about that one. So if you're looking at mRNAs, that means you're really looking at that transcriptome. So the 3' RNA-seq kit would give that kind of information for your mechanism of action for your drug. So you could look at how that drug is affecting all of the different cells in your sample in that single cell level of the transcriptome.
Awesome, thank you. Now, are you using only live cells to encapsulate?
Yeah, I can answer that one. So yes, actually, thank you for asking that question, because one of the things that I wanted to bring up is that for you to be able to generate high quality data, you wanted to make sure that yes, the cells are alive and that the quality, like the cells are happy and healthy, essentially, right? Because it is single cell. What we would want is for the samples that we are inputting of the highest viability, and that they are single cells and they are free from aggregates.
Thank you, Errile. Looks like we have time for just a few more here. Well, one of them is just more of a comment. I wanted to tell you, wonderful session. Congrats and thanks. So kudos to you both from someone in our audience. I'm going to go ahead and say that this is going to be our final question, unless there's any other audience members that chime in during the answer here. But is the nuclei isolation step required to do single cell ATAC-seq?
I can answer that one. Yes and no. Like I mentioned in the presentation, we have the two different tagmentation processes. We have the OmniATAC tagmentation protocol and the whole cell tagmentation. The nuclei is required to be accessed as part of the technology, but the two different tagmentation process, the OmniATAC tagmentation is where there is a separate nuclei step for that. In the whole cell tagmentation protocol, the cell is permeabilized, so then you can access the nuclei, but there is no separate nuclei isolation step for that.
All right. It looks like we did get another question in. What is your bead size?
I can take on that one. I don't know off the top of my head, but I can certainly like reach back for that information.
Okay, perfect. And one final question here before we wrap up. Have we tried starting with frozen cells?
Yes, we do have information on that. We have a resource on the website for cell preparation guide that has the protocol for thawing from frozen cells. Internally, we have done it on frozen PBMC, and so we have information and data, for compatibility on that sample type.
Perfect. All right, well, let's go ahead and wrap up here. Errile and Patricia, thank you so very much today for your time and your important research. Now, any questions that we didn't have time for today and those submitted during the on-demand period will be addressed by our speakers via the contact information that you registered with. Now, this webcast can be viewed on demand for 1 year until November 20th, 2025, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Now, in just a moment, you will see a survey appear on your screen. We highly encourage you to take a second to complete that survey, as it is important for us to gather that data to make sure that we're always bringing you the best content and experience. Thanks again to our audience for joining us today, and again to Bio-Rad for bringing us this webinar. I hope you all have a great rest of your day, and we will see you next time. Goodbye, everyone.